All right, so here we are in the second part of 17.4, initiation, elongation, and termination. So let's talk about initiation. The big idea of initiation is just the pieces coming together. So first we're going to have the small subunit is going to come together with the tRNA and the mRNA. Later on, the large subunit is going to attach. And once the large subunit attaches, then initiation is complete. But let's talk about the steps that have to happen during this process. So the first step in translation is the small subunit, so that's this guy right here, is going to bind to both the mRNA and the first tRNA, which I guess would be here, right? And this tRNA is going to be carrying methionine, or the amino acid methionine, which is going to correspond to the codon for start, the start codon. And so in bacteria, the small subunit can bind to either in either order. So it can bind to the tRNA or the mRNA first. They come together in whichever order. So here's the binding site for the small subunit and the mRNA. They come together, and then it scans down. So the mRNA is going to sort of move downstream until it reaches, until the A site reaches the methionine, or the start codon. In eukaryotes, it's a little bit more stringent, and the tRNA first has to bind to the subunit. So the first tRNA is going to bind to the small subunit. Then the mRNA is going to bind right here to the small subunit. But it's going to first, it needs to bind to the, attached to the 5' cap. So remember, in eukaryotic cells that we had to create our primary script, which included a 5' cap, that modified guanine at the end. So it's going to attach to that, and then it's going to move downstream until it reaches AUG, which is going to correspond to methionine, or to the start, which is what we call the start codon. Then the tRNA is going to hydrogen bond to the start codon, to the AUG, and then we're ready for the large subunit. So here's our first part, our start codon, the mRNA, and our first tRNA. Here's our AUG. Everything's lining up. And then part 2, the large ribosomal subunit, is going to attach, is going to be at the expense of a single GTP, and they're going to be initiation factors that are going to help to facilitate this this assembly here. And so at this point, the methionine is going to be sitting in the P site. So when the large subunit comes to the game, then this first tRNA is going to be sitting in the P site, ready to start the amino acid chain. And so from here on out, when the amino acids or the tRNAs enter the A site, they're going to be coming here and then adding to the carboxyl end. And we talked about this before. The amino acids are going to be added to the carboxyl end. And so now we have finished initiation. Let's start talking about elongation. Elongation happens in 3 steps. The first step is codon recognition. And we've kind of talked about all of this, so it'll come together pretty easily. Codon recognition is when the tRNA goes into the A site. And remember, we said that sometimes it isn't a match, and so the tRNA goes away, and then the right tRNA comes along. And once the right tRNA comes along, the hydrolyzation of a GTP, or at the expense of a GTP, the tRNA is going to fit more snugly and more efficiently to the mRNA. So now we're at step 2 of elongation. This is where we're going to form our peptide bond between our growing strand here. We're adding to the carboxyl end. But we were in the P site, but while we were still in the A site, the amino acid was added to the carboxyl end. So when the tRNA joined the A site, it added to the carboxyl end. The amino acid was added. Then it was translocated. The tRNA was translocated to the P site. And at this point, we have a growing chain attached to a tRNA. And so here, when it's in the P site, this is when we break that bond between the tRNA and the amino acid. So in the A site, we tied it to the growing strand. In the P site, we cut it from the tRNA. And so this cut here, this tRNA, is actually eventually going to be cut. The last one is going to be cut by a water molecule. So let's think hydrolysis when the last amino acid is added. But we'll think about that when we get there. Finally, translocation. The tRNA that's in the P site is going to be sent over to the E site, which exists only in the large subunit. And once it's in the E site, it's going to exit. And so once it exits, then the P is going to move to the next site. The A is going to open up. And so we're just saying here that the third step is translocation, that we're going to move that tRNA from the P site to the E site and open up that A site for the next amino acid, or for the next tRNA to find the codon. So this is elongation. Let's now talk about termination. Elongation is going to continue until it reaches a stop codon. Remember, we said that there were 3 stop codons. There aren't amino acids for a stop codon. Remember that the stop codon is a signal. It is a signal to stop translation. And so no amino acid is added. What is added is a release factor. So here we are at the last tRNA, and we've reached a stop codon. The stop codons are UAG, UAA, or UGA. And when we read one of those codes, the release factor is going to bind, and then it's going to release a water molecule right here. Remember, we were talking about that. It's going to release a water molecule to the growing end of to the carboxyl end of the growing chain, which is going to hydrolyze the bond, or it's going to release the strand. So remember, hydro, adding water, hydrolysis, to break. So the amino acid is released. The 2 subunits then dissociate, so they are going to break apart, and this is done with the assistance of 2 GTP molecules. So we have gone through initiation, elongation, and termination. Now let's talk about post-transitional modifications. What happens to this protein after it leaves the ribosome? And so while amino acids are being assembled, they're going to immediately start to coil. We have learned about primary, secondary, tertiary, quaternary structure. This is what's going to happen. Those intermolecular forces, those hydrogen bonds, are going to start to interact, and they're going to start to form their 3D shape. Amino acids are going to sometimes require sugars, lipids, and phosphate groups. Sometimes they're going to be cleaved in different spots. Sometimes the protein chain is going to be cleaved and broken apart, and sometimes there's going to be quaternary structure, so those polypeptide chains are going to need to come together. And so this is going to happen after it leaves the ribosome. And so there are 2 places that we're going to find ribosomes: in the cytosol, and then also in or attached to the endoplasmic reticulum. But the story always starts while it's in the cytosol. So the growing polypeptide chain, or the polypeptide chain, is going to always start to be released into the cytosol from a free-floating ribosome. But if a signal is received, there's going to be a signal on the protein. If it is received or found, then the ribosome is going to move towards the endoplasmic reticulum, and then it will be bound. It's going to be bound and release its protein into the endoplasmic reticulum. This signal peptide is only found on proteins that are going to be released out of the cell. So this should make a little bit of sense. If a protein is going to be released into the cytosol, then the protein is going to stay in the cell and do jobs within the cell. But if this protein needs to leave the cell, it's going to need to have a vesicle around it, because it's going to need to be able to attach to that plasma membrane to leave. And so here it's going to enter into the endoplasmic reticulum. It's going to be modified, and then it's going to exit out, and it's going to have that vesicle attached to it or surrounding it so that it can leave the cell. So an example of that would be like insulin. Insulin is made inside the cell, and then it's released into the bloodstream so that it can help to take sugar up in our blood. And so let's talk about this signal peptide. It is a sequence of about 20, it is a sequence of amino acids, about 20 amino acids from the N-terminus. So once the amino acid is being made, about 20 amino acids in, you're going to have it's called an SRP, the signal recognition particle. So here's our mRNA going through, being read by the ribosome, and our protein or our polypeptide chain is being synthesized. And it's saying that the signal, the SRP, or the signal, is right here, and so the factor, or the SRP, is going to be attached to that growing strand, and it's going to lead the ribosome to the endoplasmic reticulum so that the chain, the growing chain, can be deposited into the endoplasmic reticulum, or to the lumen of the endoplasmic reticulum. And so therefore it will have a vesicle for transport to its destination. Finally, here's our last slide. We've made it. In both bacteria and eukaryotes, a ribosome can synthesize an average polypeptide, so an average protein, in less than a minute, which is crazy and very cool. But we can increase the rate at which proteins are made by increasing the number of ribosomes that are working on a single strand of mRNA. So once the mRNA is far enough out of the ribosome, another ribosome is going to attach onto it and start working on it. So you're going to have a protein being released here, a protein being released here, a protein released being here. This is called a polyribosome. So you can see multiple ribosomes attached to this one mRNA strand. And so this is going to help to express a trait more quickly. And this is gene expression. So this is the end of 17.4, and we have 17.5 next. Okay.